I'm Timo Laaksonen. I'm a professor of pharmaceutical nanotechnology at the University of Helsinki and Faculty of Pharmacy. And I'm running a research project and research team focusing on drug delivery with nanoparticles, liposomes, and light activation. So we are making photosensitive liposomes for on-demand drug release applications. They can be sensitive to light in terms of that they generate heat or radical oxygen species. But in terms of this photothermal liposomes, what we do is we have dyes that absorb light, specific wavelength, and generate heat, and our heat-responsive liposomes that release drugs. The main challenge might be how well you can localize the effect. What you want to do is you have a system where nothing is released at all without activation, and then it's sensitive to, for example, red light, and then the red light, when it's focused in exactly the same place where the liposomes are, then releases the drug. Light for us is helping to localize the drug release in specific parts, for example, specific tissue, specific organ, specific cells. The ML8500 is used for us because it's a plate reader setup, and then we have a lot of samples. We do screening, so we need to find what formulation works best. So we end up running a lot of samples. So we want to keep our samples always on the same plate, just move it from one machine to the next. And with this one we can do it, and then that allows us to run a lot of samples. We run a screening study of many different kinds of liposome formulations. There were dozens of different samples that we tried. We found out a good formulation that was not releasing the drug in a passive manner, was heat sensitive, and was sensitive to light.